Hello students, welcome to this particular module. In this module, we are going to discuss about the general characters and classification of pteridophytes. The pteridophytes are regarded as the oldest vascular plant that colonized land during primitive time. They are seedless plant and bear no flowers, and hence they are regarded as cryptogams or non-flowering plants. They are adapted to land habit and thus show some distinct features. Talking about the characteristics of pteridophytes, first comes is spores. The spores of pteridophytes are two-layered. One is the inner layer known as the intine and the other was the outer layer known as the exine. These layers protects the spore from drying up. As a result, these desiccation-resistant spores can travel to distant places. You can see in this picture, the outer layer is the exine and this inner layer is the intine. The second is the presence of cuticle and the stomata. The epidermis of the exposed part are provided with a thick layer of cuticle. The cuticle protects the plant from drying up. That is, it helps in retaining the water inside the body of the plant. You can see here I have given three pictures. One is of the root, the second is a stem and the third is the leaf. In the first one, in the root, no cuticle is seen, but in the exposed areas, that is the stem and the leaves, have the cuticle and in the outer layer of the epidermis. Now, since cuticle is present, it hinders with the exchange of gases between the plants and the external environment. As a result, stomatas are there which help in gaseous exchange. Here is the structure of the stomata. The third are the tubes and the tracheids. This help in the conduction of the water. The conducting system was essential as the plants grew to the erect habitat. Slowly with time, these tracheids become lignified, thus increasing the fluid conductivity and also decreasing the permeability. Here I have given a picture in which you can see that how this tracheids is being formed from simple water conducting cells and how slowly only from primary cell wall composed of cellulose, a secondary cell wall was formed that is composed of lignin. Coming on its habitat, the pteridophytes are usually terrestrial. They prefer to grow in moist, shady and cool places. Some of the pteridophytes are aquatic, for example Azolla. Some are even found in desert conditions, that is in water-stressed condition, for example Selaginella, and some are even found as epiphytes growing on tree trunk, example Lycopodium. Now we can divide, if we are talking about the characteristics, we can divide this characteristic based on its sporophytic characters and its gametophytic characters. First, we will be talking about the sporophytic character. The plant body is divided into root, into stem and leaves. Although in some primitive pteridophytes, true root are not present. The sporophyte develops from a diploid zygote. Firstly, talking about leaves, in pteridophytes we get two different types of leaf. One, the microphyllous leaves and the second is the megaphyllous leaf. The microphyllous leaves are simple, the midrib is single and unbranched and no leaf gap is present. For example, the leaves as seen in Lycopodium. In case of megaphyllous leaves, the leaves are large, compound. The midrib that is present is dissected and prominent leaf gaps are there. For example, Marsillia. In this picture, in the first picture, you can see that how these microphyllous leaves are being formed from the naked central axis. So this is according to the annation theory. Here we have a picture of the megaphyll. These are the megaphylls in which you can see the midrib is dissected and the leaves are compound and it is comparatively larger as compared to the microphyle. Now coming to its internal anatomy, we can see that pteridophytes have a prominent vascular bundles. The xylems and phloems are present, but there is no cambium in between these two. That is, no secondary growth is usually seen in most of the members. The stele is very diversified in case of pteridophyte. In primitive member, we see protostele. Slowly with evolution, siphonal stele is being seen. In certain cases, in the same species also, two different types of steles can be present. The sporangium in all pteridophytes is initiated by division of the superficial cell or sometimes a group of cells. In this regard, we can have two different type of mode of development. First is known as eusporangiate and the second is known as leptosporangiate. If the sporogenous tissue is derived from several sporogenial initial, then it is known as eusporangiate, and this occurs in most pteridophytes. If it develops from a single initial, then it is known as leptosporangiate, and this is a feature of advanced pteridophytes. In this picture, you can see that in the first case, that in the eusporangiate development, a group of cells are involved which are acting as the sporogenial initial. But in the second case, only a single cell is present, and in this too, the ultimate thing is the sporangium. In most pteridophytes, these sporangia are aggregated to form a cluster-like structure, and this is known as the sori. This sori can be of three types. The first is the simple sorus, the second is the graded sorus, and the third is known as the mixed sorus. In case of the simple sorus, the sporangia originate, grow and developed all at the same time. For example, as seen in Ophioglossum. When the sorus developed at different time and the central portion contains mature sorus, whereas the peripheral ones are the younger. Example, Marsilea. And the last one, when the sorus develop at different time and mature, the immature and mature sporangia are arranged in irregular pattern is known as mixed sorus. For example, Pteris. If you can see this picture, in the first one, this is the simple sorus, where all these sorus, as you can see, are present in the same line. Here, the middle one is the matured one, whereas these are relatively younger. But here, they are irregularly arranged and there is no definite pattern of formation. The spores are produced after meiotic division and numerous haploid spores are produced inside the sporangia. When the spores are of similar shape and size, these are known as homosporous. For example, Marsilea. If the spore produced are similar in size, but in certain condition, if the females are not fertilized, it may act as the male. This condition in which although all the spores are alike, but 50% of it is forming the male and 50% is forming the female, and again this female, if remain unfertilized, can act as the male, is known as incipient heterospory as seen in Equisetum. And this third type is known as heterosporous, where two different spores are produced. One is the megaspores and the other is the microspores. These megaspores are relatively larger and the microspores are relatively smaller. These megaspores form the female gametophyte and the microspore form the male gametophyte, and this particular phenomena is seen in Selaginella. Here in this picture you can see that the megaspore is comparatively very large as compared with the microspores. So this is the homosporous condition when isospores, just simple isospores are produced, and it has evolved to produce two different types of spores, the megaspores and the microspores. Now here I have just given the alteration of generation that I just talked about, that isospores will be forming a symbol gametophyte, that will be bisexual. But in case of heterosporous condition, two different gametophyte would be produced, the microspores producing the male and the megaspores will be producing the female. Now this germination of the spores can be of three types: bipolar, tripolar and amorphous and irregular. The development of gametophyte is again are of two types, exosporic and endosporic. This first one is exosporic. It says that the prothallus develops outside the spore wall. If we consider this to be the spore wall, so here the prothallus is getting developed outside the spore wall. For example, xylem. In case of endosporic, the development of prothallus is inside the spore wall. If this brown line, this outer covering is the spore wall, and inside this brown covering we can see that the development of prothallus is taking place, and this is completely independent of the external environment. Example, Selaginella. Now coming to the gametophytic phase. The gamete bears both the sex organ, the male and the female. The male known as the antheridia and the female known as the archegonia. You can see this archegonia is present just below the apical notch, whereas the antheridia are present as the lower portion, and it is usually amongst the rhizoids. The archegonia that is being present is a flax-shaped structure, and it consists of a lower venter and a short neck. This venter encloses an egg and the ventral canal sac. The neck is formed of the neck canal cell. Coming to antheridia, the antheridia are usually short or sessile and are globose in structure. Inside it is present the anthrozoids, which are unicellular, uninucleate and spirally coiled, and it bears the flagella. In pteridophytes, the male is motile and it swims towards the female, that is towards the egg. In this respect, you can see these are the neck cells. These neck canal cells are disintegrated when the archegonium attain maturity, and on this disintegration some sort of chemicals are being released. Malic acid or fumaric acid are released, which act as attractant, and with the help of this attractant the sperms are attracted towards the egg, and as a result fertilization takes place. After fertilization, an diploid zygote is formed by the fusion of the two haploid gametes. On the basis of the polarity, the embryo can be categorized into two types: exoscopic embryo and endoscopic embryo. In exoscopic embryo, the shoot-forming apical cell is directed towards the neck of the archegonium, that is directed outward. Endoscopic embryo, the shoot-forming apical cell is directed downwards, that is towards the base of the archegonium. Now the basic life cycle pattern of pteridophytes shows a regular heteromorphic alteration of generation between a gametophytic phase and a sporophytic phase. The sporophytic phase is the dominant phase in the life cycle of pteridophytes. Now we will be talking about the classification of pteridophyte. In primitive time, the classification was based on the information that we got from the natural system of classification. In primitive time, the plant kingdom was divided into Thallophyta, Bryophyta and Pteridophyta. If you can look in this, you can see this is the whole plant kingdom. We are having two parts. One is the cryptograms and one is the phanerograms. Various classification was proposed during different times, but some notable amongst them are Ames 1936, Tipo 1942, Smith 1955, Cronquist 1960, Frost and Gifford 1974, Knoll and Rothwell 1981, Stewart and Rothwell 1993. Jeffrey in 1902 divided the vascular plants into two stocks. In the first stock, he kept ferns, gymnosperms and angiosperms and termed these as Pteridopsida, and in the second stalk he kept lycopods and hostels and known as Lycopsida. He could not name all this stock. Later on in 1923, Scott ranked them as division and included hostels as a separate division, the Sphenopsida. Ames and Tipo considered a single division, the Tracheophyta, and divided them into Psilopsida, Lycopsida, Sphenopsida and Pteridopsida. Actually, by Tracheophyta we mean to say all the plants that have the tracheary tissue, that is xylem, and all the plants having this particular tissue are known as Tracheophyta. Tracheophyta are regarded as a major taxon and is a monophyletic origin of vascular plants. Here you can see the classification. You can see here there is two columns. One is this column and the second one is this column. Both of this classification was given by Gifford and Foster in 1996. In the first one, they included Tracheophyta as the division and the rest were the class. But the later on, with some revisions, they say that this were the divisions. That is, they brought this class to the division. You can see here. Rhinopsida is turned into Rhinophyta, which comprises of extinct plants, that is Rhynia cooksonia. Zoosteropsida, Zoosterophyllophyta. It has extinct plant, example Zoosterophyllum. Trimeropsida, Trimerophyta. Extinct plant Xylotum and Trimerophyton. Psilopsida, Xylophyta, having two living genera, Xylotum and Mesipteris. Then coming to Lycopsida, again Lycophyta, it have some extinct forms as Prolepidodendron, Lepidodendron and some living genera like Lycopodium, Selaginella, Xyloglossum, Isoetis, etc. Sphenopsida to Sphenophyta, consisting of some extinct plant like Calamites, Sphenophyllum and one living genera, that is Equisetum. And the last one, Phelicopsida, which was then again transferred to Phelicophyta. It comprises of the living as well as the extinct fronds. Coming to each group, we will see. First one is the Rhinophyta. Rhinophyta includes members which have simple sporophytic morphology and reproduction is also simple. The rhizomatous stem bears the rhizoids. It is a leafless, naked axis and each axis is terminated into the sporangia. This is the reconstructed plant genus. Second one is Zoosterophyllophyta. It has been dichotomously branched, having a rhizome system, and the sporangia appears to be colline. That means the sporangia are present in the stem. It is attached in the stem like this. The third one is Trimerophyta. The plant has stout main axis and dichotomously branched lateral axis. The axes are provided with blunt spines and it has interrupted longitudinal ridges. The sporangia are elongated and elliptical. You can see this particular picture of Trimerophyta. Another thing, all these are homosporous in nature. Coming to the second one, Xylophyta. Here also it is dichotomously branched and the stems have relatively simple vascular cylinder. The sporangia are eusporangiate and are present at the end of the shortened axis. You can see that it has a shortened axis which bears the sporangia. Coming to Lycophyta, the sporophytic plant body is now differentiated into leaf, stem, root and sporangium, which is a eusporangia. The microphyls that are present can be ligulate, that is a ligule may be present, or euligulate. Each sporophyll has one sporangium attached with it. The vascular bundle can be protostelic or siphonostylic and there is no leaf gap. exarch xylem predominates. So here is the picture. You can see the stem, leaves, root. It is present in this particular group. In Sphenophyta, the sporophytic plant body is again differentiated into stem, leaf, root and eusporangia. Here we see there is distinct node and internodes, and in each node we have simple scale leaves. Xylem is exarch or endarch, and secondary growth is present in some members. Sporangia here are borne on specialized structure known as sporangiophores. These sporangiophores are again clustered to form a structure known as strobili. They are usually homosporous, but in some members heterospory are also seen. So here is the picture. We have different species of this particular group. Last we come to Filicophyta. This is the most advanced among the pteridophytes, and here the leaves that are present is the megaphyll. They are large and they have a branched venation system. Here leaf trace and leaf gap is prominently present, and if in the vascular system if it is not protostele. So it has attained the highest mode of organization in case of its leaf structure. It is rashes is present in this particular members, and the sporangia can be both eusporangiate or leptosporangiate. The collection of sporangia here is known as sorus, and this sorus in some genus is protected with the help of a particular structure known as the indusium, and the sporangia of higher ferns are present on the adaxial side of the fertile pinnae, and the adaxial sporangia frequently fuse to form a structure known as synangia. So here we end with the classification of pteridophyte. So first we learnt about the various characters in general about the pteridophytes, and second the classification of the pteridophytes.